Welcome students. My name is Ms. Karen Lenisha Cabral, Assistant Professor in Botany at St. Joseph Vaz College, Kottali. This program is for Bachelor of Science, second year, subject Botany, semester 4, course code BOC 104, course title Plant Physiology. Title of the unit Enzymes, module name Properties of Enzymes, Part 1. Outline, Introduction, General properties of enzymes, specific properties of enzymes, catalytic property, specificity and reversibility. Learning outcome describes the properties of enzymes. Now, enzymes are biocatalysts of proteins in nature. Now, they accelerate the rate of biochemical reaction but does not affect the nature of the final product. They regulate the speed and specificity of reaction without being used up, but unlike catalysts, enzymes are produced by the living cells only. Now, when we are talking about living cells only, we are referring to the biocatalyst, because when we say catalyst, it can also be a chemical catalyst that we are referring to. But the moment you say biocatalyst, it wholly belongs to, or it means enzymes. And enzymes are produced only in living cells. Now, enzymes influence the rate of biochemical reaction, so they can take place at a relatively low temperature. Thus, enzymes are known to lower the activation energy. Now, the term enzyme are derived from Greek word which means in yeast, because the yeast cells were first to reveal enzyme activity in living cells. Now, the term was coined by Kuhne in the year 1878. Coming to general properties of enzymes. Now, enzymes initiate and accelerate the rate of biochemical reaction. Now, this is because they are biocatalysts, without which the process would have taken a way longer time. Now, the activity of enzyme depends upon the acidity of the medium, that is, it is most active at a specific pH. Now, enzymes can accelerate the rate of reaction in either direction, and all enzyme possesses an active site which participate in the reaction. Now, this active site could be more than one. Now, enzymes are very unstable compounds, mostly soluble in water, dilute glycerol, NaCl, and dilute alcohol. Enzymes act actively at optimum temperature. The moment enzymes are subjected to higher temperature, their activity drops drastically. All enzymes are protein, but all proteins may not be an enzyme. Enzymes lower the energy of activation of the substance molecule so that the biochemical reaction can take place at normal body temperature. Coming to the important properties of an enzyme, we are going to touch upon, we'll see three of it. One is the catalytic property, second one specificity, and thirdly reversibility. We'll look at each one in detail. The first one being catalytic property. They have extraordinary catalytic power, that is with regard to the rate of the reaction. They are active in very small quantity. Now, small amount of enzyme converts a large quantity of substrate. Enzyme remains unchanged after the reaction has been completed. Now, the turnover of enzyme ranges from 0.5 to 6 lakhs. Now, what exactly is a turnover number? It is the number of substrate molecules converted by one molecule of enzyme per second when its active site is saturated with a substrate. Coming to the second property, that is specificity. Enzymes are very specific with their action. Particular enzyme act on particular substrate only. Now, enzymes are also specific to particular type of reaction. Enzymes show different types of specificity. Here we have 6 which are jotted down. First one is bond specificity, group specificity, substrate specificity, optical specificity, cofactor specificity, and geometric specificity. Now, when we come to the first one, that is bond specificity, it is also called as a relative specificity. Now, the enzymes are specific for a particular bond. For example, you have peptidase, an enzyme which is specific only for a peptide bond. Then we have group specificity. Here the enzymes are specific for a group. Example, here we have pepsin, which hydrolyzes the peptide bond in which the amino group belongs to the aromatic amino acid. Third one, substrate specificity. The enzyme acts only on a particular substrate. Example, arginase, which acts only on arginine. Carbonic anhydrase acts on carbonic acid. Then the fourth one being optical specificity, it has the highest specificity, and the enzymes are specific not only to the substrate but also to the optical configuration. Example, you have L-amino acid oxidase which acts only on L-amino acids. Then we have cofactor specificity. This shows that enzymes are not only specific to a substrate but also very specific to the cofactor. And the last one is geometric specificity, which is less specific. Now, some enzymes work with small range of similar substrates having similar structural geometry. Example, alcohol dehydrogenase can oxidize methanol and N-propanol to aldehydes. Then the third important property is reversibility. Most of the enzyme catalyzed reactions are reversible. Now, the reversibility of the reaction depends upon the requirement of the cell. In some cases, there are separate enzymes for forward and reverse reactions. Some enzyme catalyzed reactions are not reversible. These are my references. Thank you.